Good morning. My name is Martha Donoghue, and I'm a pediatric hematologist-oncologist and serve as the Deputy Director of the Division of Oncology II in the Office of Oncologic Diseases in the Office of New Drugs at the FDA. This morning, I'm going to provide a brief presentation on the design of first-in-human trials for small molecules and biologics. The aim of this presentation is to give you a better understanding of the important key design elements of first-in-human trials. I hope you also gain some insight regarding next steps for clinical development beyond the initial dose-finding component of a first-in-human trial. And finally, I will give you an inside glimpse of how the FDA reviews new INDs containing first-in-human trials for the development of cancer drugs and biological products, which I will simply call drugs from here on out, and relay some tips to help you successfully navigate the process for submitting your original IND application for a first-in-human trial and increase your chances of receiving a study may proceed letter the first time around. There are several important trial design aspects to think about when formulating a protocol for a first-in-human trial. These include the patient population, dose-finding design, safety monitoring plan, risk mitigation measures, and other study provisions such as pharmacokinetic, pharmacodynamic assessments, and measures of response to treatment that provide key information needed to make decisions regarding whether and how to study a drug in patients with cancer in future trials. There are also other important documents that are needed for the safe and ethical conduct of first-in-human trials: the informed consent document and investigator brochure, if it's a multi-center trial. However, before thinking about these individual aspects of first-in-human protocol design, it is important to take a step back and think about the big picture. In other words, as Stephen Covey, the author of 7 Habits of Highly Effective People, famously said, begin with the end in mind. For a first-in-human trial, as in all aspects of drug development, the foundation should rest on the deepest possible understanding of the drug, its purported mechanism of action, how this mechanism of action is likely to exert an effect on tumors, and what the off-target effects are likely to be. This knowledge will help you determine which patient population you would like to include in your first-in-human trial and the protocol provisions that will be needed to preserve their safety. Additionally, and I know this seems obvious, but I still think it is worth stating, before sitting down and writing a protocol, think about and identify what your goals for the trial are and what information you will likely need to decide whether and how to move forward in clinical development for the drug, including the patient populations that you think are likely to benefit most from treatment. A word of caution, stick to the objectives that you think are most vital and are achievable, and don't overload your study with a lot of exploratory objectives that are not likely to aid in decision-making. This will help ensure that the trial can be conducted efficiently and will ultimately provide the information you need for future decision-making. Then, design the trial accordingly, assuring it is well-organized, internally consistent, and clearly communicates the information about your drug, your goals, and the overall approach to ensuring these goals are safely met. Irrespective of the type of drug, the objectives of a first-in-human trial are generally threefold and typically are achieved through conduct of a 2-part trial. The first part of the trial generally consists of a series of steps designed to make a preliminary evaluation of the toxicity of the drug in patients with cancer and identify the maximum tolerated dose, or MTD, or optimal biologic dose in patients with cancer, in cases when reaching an MTD may not be necessary or appropriate. And ultimately, to identify a recommended dose for future evaluation, which is generally termed a recommended phase 2 dose, or RPD. After the initial dose-finding portion of the trial is complete, most first-in-human trials have a second phase 2 component, in which the safety and anti-tumor activity of the drug is further evaluated at the RP2D, generally through small expansion cohorts which comprise patients who have a particular type of cancer or who have cancers that can be identified through a common biomarker. Anti-tumor activity is generally assessed through assessment of overall response rate and duration of response, although other important pharmacokinetic and pharmacodynamic assessments may also be performed. The eligibility criteria for first-in-human trials identify the patient population that will be enrolled. Most commonly, first-in-human trials enroll patients with cancer, not healthy subjects, although occasionally first-in-human trials will evaluate a very low dose of a candidate anti-cancer drug in healthy subjects. During this talk, I will focus on first-in-human trials that enroll patients with cancer. In general, the eligibility criteria should limit enrollment to patients who do not have curative options. In other words, those who have a serious, unmet medical need. Typically, eligibility criteria for a first-in-human trial restrict enrollment to patients who have already received treatment with available approved therapies known to be effective for the treatment of the disease under study. In the non-curative setting, some sponsors prefer not to enroll heavily pretreated patients who have exhausted all available treatments, and this is acceptable when there is appropriate scientific justification to do so, particularly when these available therapies do not confer a large magnitude of benefit. In these cases, we ask companies to include information in the informed consent document that by enrolling on the study, they may be foregoing available therapy with a proven benefit. The eligibility criteria should also aim to restrict enrollment to patients who are not at increased risk for serious toxicities related to the drug, based upon the available information and non-clinical studies identifying the most likely toxicities that patients will encounter and the drug's mechanism of action and anticipated route of metabolism. In general, eligibility criteria include provisions that limit enrollment to patients that have adequate organ function and do not have comorbidities that will place them at increased risk for a life-threatening event. It is also important to include criteria addressing the timing of enrollment with respect to prior treatments. Lastly, eligibility criteria generally indicate whether special populations, such as pediatric patients, may enroll. One additional thing to consider relates to targeted therapies. If the drug is unlikely to benefit patients whose tumors or tumor microenvironment does not harbor a specific biomarker, then consider including eligibility criteria restricting enrollment to patients with this biomarker, and including a requirement that there be sufficient tumor tissue for initial and future testing. We at FDA have worked for the past several years with stakeholders such as ASCO to provide recommendations to ensure that eligibility criteria are not unduly restrictive so that studies can enroll patients that more closely reflect the patients who would receive the drug in the post-market setting and provide more equitable access to clinical trials. There are multiple FDA guidances on this, and I have included some in the resources section at the end of this presentation. First-in-human protocols pre-specify a definition of dose-limiting toxicities, or DLT, which are toxicities generally considered to be severe enough to prevent dose escalation when they occur at an unacceptable frequency. These DLT criteria guide dose escalation and de-escalation decisions and are also used to identify the maximum tolerated dose, or MTD. In general, these DLT are severe treatment-emergent adverse events that occur during the DLT window, which is usually one cycle of treatment, typically 4 weeks. Although treatment-emergent adverse events that are at least possibly related to study treatment during that time and are severe are generally considered DLT. Severity of treatment-emergent adverse events is graded using the NCI Common Terminology Criteria for Adverse Events, or NCI-CTCAE, and most treatment-emergent events of grade 3 or greater severity, with a few exceptions, are considered DLT. Having said that, the DLT definitions and the length of the DLT evaluation window do vary depending on context. For example, DLT would be defined much more conservatively in a healthy volunteer study, and we may be a little more lenient in the DLT definition in patients who are acutely ill and are expected to receive treatment in a highly controlled hospital setting. For drugs with long half-lives, an extended period of observation beyond one cycle may be needed. Most first-in-human trials employ a dose escalation design starting at a relatively low dose with incremental stepwise increases in doses if the current dose level does not cause undue toxicity. There are two basic types of approaches to dose escalation: a rule or algorithm-based approach, or model-based approaches. The 3+3 design is the most common, simplest, and traditional approach. With this design, 3 patients initially enroll in a cohort, and if a single dose-limiting toxicity occurs, the cohort expands to include up to 3 additional patients. If 0 of 3, or no more than 1 in 6 patients in the cohort has a dose-limiting toxicity, then enrollment into the next highest cohort begins, and so on until a cohort is reached in which more than 1 patient exhibits a DLT, or until the maximum dose intended for study is reached. There are alternative rule-based approaches, such as those including accelerated titration. There are multiple types of accelerated titration designs. One example enrolls patients into single-patient cohorts until toxicity above a certain level is observed and then reverts to a 3+3 design. As I mentioned earlier, model-based designs are alternatives to rule-based designs. They use a statistical model to estimate the relationship between the dose and DLT risk, which then informs dose escalation decisions. The model is also used to identify the maximum tolerated dose, which is defined relative to a target toxicity level specified by investigators before the trial begins, which is generally less than 33% of patients. There are several types of model-based methods that can be used, some of which are quite complicated. The modified toxicity probability interval method is one of the simpler model-based designs, but the continual assessment method, or CRM, and escalation with overdose control, EWOC methods, are also frequently used. To go into these methods are beyond the scope of this talk, but there is a body of work supporting model-based designs as more accurate and efficient in identifying the MTD compared to traditional rule-based approaches, such as the 3+3 designs. And designs such as CRM have the potential to assign more trial participants at or close to the MTD compared to rule-based approaches such as the 3+3 design. Irrespective of which dose escalation design is used, I generally recommend that a safety committee review the accumulated safety data, including adverse events that occur outside the DLT monitoring period, prior to making the decision to dose escalate. And this information should also be used when identifying the MTD and recommended phase 2 dose. When thinking about the dose levels to investigate for first-in-human trials, we rely heavily on information from the pharmacology toxicology studies to identify a starting dose that has an acceptable safety margin, and also takes into account any additional information that is available for drugs with similar properties that have been previously studied in humans. There is a balance here between starting at a dose level that is safe, but not unnecessarily low, because we also want to limit the number of patients exposed to a subtherapeutic dose. It is also important to identify the appropriate dose increases to make between subsequent cohorts. In general, doses for biologics are increased by half-log increments, or a factor of 3, whereas for small molecules, there is generally at most a 100% increase between dose levels. For small molecules, successive dose levels can be increased by fixed increments or by varying decreasing percentages, such as a modified Fibonacci design. Factors to consider when thinking about how much of a margin to increase doses by for small molecules include the expected dose toxicity relationship that was observed in animal studies. If this dose toxicity relationship is relatively flat, then more aggressive dose escalation can be supported, whereas if the dose toxicity relationship appears steep, then a more conservative approach is warranted. In some cases, including provisions for intrapatient dose escalation is appropriate, which allows patients who have tolerated a lower dose level for a certain number of cycles are allowed to increase the dose to a higher dose level than has already been studied and determined to be safe. Before discussing other aspects of first-in-human trials unrelated to dose finding, I would like to emphasize a few key principles. First, it is not always necessary or justified to keep dose escalating until a maximum tolerated dose is reached. In the case of non-cytotoxic therapies, it is smarter and safer for patients if the goal is to identify the minimum biologically effective dose based upon available safety, tolerability, efficacy, PK, and pharmacodynamic data. So the recommended phase 2 dose and maximum tolerated dose are not necessarily the same things. Additionally, the MTD technically reflects the toxicities that occur early during dosing, generally during the first 4 weeks of treatment or so, and the recommended phase 2 dose should also take into account overall tolerability of the drug, including the toxicities that occur beyond the DLT period. Furthermore, we now have the patient-reported outcomes version of the CTCAE criteria, which can provide valuable information regarding the patient's perspectives on toxicity. We encourage sponsors that they consider using this patient-reported assessment tool in addition to investigator-recorded adverse events and grading of adverse events as an additional way to help paint a clearer picture of the safety and tolerability of their drugs to help with identification of the appropriate RP2D. The dose you take to the dose expansion phase of a first-in-human clinical trial is known as the recommended phase 2 dose, as we discussed. However, this RP2D is not necessarily the optimal dose of the drug because it is generally based on limited information, and so continued dose exploration and refinement is strongly recommended early during clinical development prior to initiating a trial intended to provide the primary basis to support a marketing application. Safety monitoring is another important component of all clinical trials, and study protocols should include a detailed schedule of events outlining the timing of patient treatment and assessments. Protocols must include explicit monitoring guidelines for safety, assessment of adverse events, laboratory assessments, physical exams, vital signs, tumor assessments, etc. For first-in-human studies, these assessments are typically fairly frequent, particularly during the first few cycles. When determining the frequency and type of safety assessments to perform, it is important to take into consideration available preclinical data for the drug and any available clinical data for the drugs with the same or similar mechanism of action, as well as the drug half-life. Aside from safety monitoring, other important risk mitigation measures are needed in protocols for first-in-human trials, such as detailed instructions regarding dose modification for toxicities, including when to hold the drug, when to reinitiate the drug, whether the dose should be reduced upon reintroduction, and when to permanently discontinue the drug in patients. The protocol should contain clear stopping rules for patient treatment discontinuation and interruption of enrollment or discontinuation of the entire study if unacceptable levels of toxicity occur despite the other risk mitigation efforts built into the protocol. Finally, protocols should describe how the overall safety of the trial will be assessed and how decision-making will occur as safety signals arise, necessitating a change in study conduct. After the initial dose-finding component of a first-in-human trial is completed, a second phase 2 component of the trial generally is opened to further explore the safety of the drug and get a preliminary assessment of anti-tumor activity. This often takes the form of multiple expansion cohorts, which can provide proof of concept to help inform future development. These expansion cohorts are generally no more than 40 patients and can utilize a variety of designs, such as the Simon two-stage design depicted in this slide. You can also include cohorts to explore anti-tumor activity in different patient populations, such as pediatric patients, if warranted, or build in a randomized phase 2 component to compare safety and activity of 2 dose levels to help with dose optimization. Next, I'll shift gears and I'll provide a brief overview of how we at FDA review first-in-human trials for cancer drugs. First, I would like to orient you a bit to our organization. These pictures are taken from our main campus in Silver Spring, Maryland, where hopefully we will return in the not-so-distant future. The Oncology Center of Excellence at the FDA fosters a unified interaction between the centers involved in the regulation of the investigation and approval of drugs, biologics, and devices for the treatment of patients with cancer. The Center for Biologics Evaluation and Research, or CDER, oversees cellular and gene therapies as well as vaccines, whereas the Center for Drug Evaluation and Research, or CDER, where I work, oversees development of drugs and other biologic treatments, such as monoclonal antibodies and antibody-drug conjugates. The Center for Devices and Radiological Health, or CDRH, oversees the development of therapeutic devices as well as companion diagnostic tests typically used for the selection of patients for molecularly directed targeted treatments. Across these centers, there are over 100 oncologists. The Office of Oncologic Diseases, or OOD, is part of the Office of New Drugs in the Center for Drug Evaluation and Research, or CDER, and comprises 5 review divisions, and each division has teams dedicated to specific cancer types, as shown in this slide. The 3 solid tumor divisions share responsibility for first-in-human trials that enroll patients with a variety of solid tumor types. The FDA role spans drug development and starts with the investigational new drug application, or IND, and sometimes even before that, which I will get into later. Our focus in the early phases of drug development is primarily the protection of clinical trial participants, and while this is always an important part of our mission, as drug development proceeds to later phases, our evaluation of the quality of scientific investigation of these drugs takes on a greater role in the work we do. Once an IND is submitted, we have 30 days to review the IND, which we generally term a 30-day safety review. During this 30-day period, we determine whether the IND is safe to proceed, and if we determine it is not safe to proceed, we place the IND on hold, which basically means that the study submitted to the IND cannot begin. During our review, we determine if a study can proceed if it does not pose an unreasonable or significant risk of illness or injury, and if the study is adequately designed to meet its stated objectives. As with most of the work we do at the FDA, the process of IND review is multidisciplinary, involving subject matter experts in non-clinical pharmacology and toxicology, product quality, clinical pharmacology and statistics, as well as clinicians. And our review is coordinated by project management staff or regulatory project managers who are specialists in drug regulation and provide the primary interface between sponsors and the FDA. Just to give you a glimpse of our internal procedures, we generally convene all of the disciplines involved in an IND review during our weekly safety meetings and discuss the IND with divisional leadership around week 3 of the 30-day IND review period. You will typically receive at least one information request via email from the regulatory project manager during our review, generally before, but sometimes after the safety meeting takes place as well. These information requests typically include recommendations for revisions to the protocol or informed consent document or requests for additional information. These generally require a fairly quick turnaround on your part within 2 to 4 days, given the tight time frame we are working under. These requests may identify deficiencies or changes that must be made in order for us to consider the study safe to proceed, or comments, which are just suggestions to think about. If you are unclear regarding any of these deficiencies or recommendations, you should feel free to reach out to the regulatory project manager with clarifying questions or proposals. At the end of the 30-day period, if you do not hear from us, the IND is safe to proceed. However, we will generally send you a study may proceed letter to let you know that we have determined that the IND is safe to proceed. If we determine that the study is not safe to proceed, we generally will arrange a teleconference with the sponsor on or before the 30-day deadline to inform the sponsor that the IND is on clinical hold and to explain the reasons why we consider the IND not safe to proceed, as well as to outline next steps for the eventual goal of fixing the IND submission so that the study will be safe to proceed and we can remove the clinical hold. The pre-IND meeting is one of the best resources available to help make sure that you are on the right track and increase the likelihood for a smooth and successful IND application process. These meetings provide an opportunity for you to seek and receive feedback on key elements in your IND application, ranging from the tox studies you plan or have performed, aspects related to manufacturing of your drug product, and/or questions related to design elements of your clinical trial. If you are interested in a pre-IND meeting, you submit a meeting request to the appropriate oncology review division along with your proposed questions for us. We generally schedule these pre-IND meetings within 60 days of the request. For first-in-human trials, these are generally written response-only meetings, and within the allotted time period, we send you a document with detailed answers to your questions, in addition to other comments and recommendations that we have based upon review of the meeting package you provide. If the topics are particularly complicated, then we will grant these meetings as teleconferences. In order to ensure that our feedback is as helpful as possible, it is important that you provide sufficient information in your briefing package for us to answer your questions. For example, if there are questions regarding the trial design, a detailed protocol synopsis or the draft protocol itself should be included in the meeting package. Additionally, the question should be as specific as possible, not merely, do you agree with our study? I hope that this presentation has provided you with some valuable information to help you design a first-in-human trial for the drugs or biologics that you are hoping to develop and help you successfully navigate the IND review process for your first-in-human trial. I would like to leave you with a few final thoughts. First of all, first-in-human trials should serve patients first. The trial should serve patients and not the other way around. The primary focus of a first-in-human trial should be to preserve patient safety while at the same time providing the chance to help them and/or future patients with cancer, while gathering important information to help lay the foundation for future drug development. Although there is no one-size-fits-all approach to designing first-in-human trials, they generally aim to provide an initial characterization of the toxicity profile and identify the recommended phase 2 dose of an investigational drug, make a preliminary evaluation of antitumor activity, and help identify patient populations and tumor types for further evaluation in future trials. Remember that dose finding is an incremental process that does not end with the dose escalation portion of the trial, and that additional dose exploration should be conducted to identify the optimal dose prior to initiation of the registration trial. And finally, I strongly recommend pre-IND meetings to facilitate development of a successful IND submission. I provided some links to valuable resources for IND applications on this slide, and links to helpful OCE guidances are listed here. I look forward to answering any questions you might have after this presentation, and thank you for your attention.
Thanks, Martha. That was strong, solid work and clinically precise. I wish all our protocols that came in could be like that. There are some interesting questions from the audience, and I'm going to run through them pretty quick. And I think the first one you kind of answered at the end, but maybe you can re-echo that answer was, first in human expansion cohorts are typically limited to 20 or 40 patients. In practice, such numbers are not adequate to compare benefit risk effectively between two doses. What approaches does FDA seem to be effective in determining an optimal biological dose?
Thanks, Jeff. That I could probably speak to for over an hour, but I'll try to distill it into a short answer. We do have that guidance out on expansion cohorts in trials in oncology that generally recommends that the size of those cohorts be limited to about 40 patients for non-hematological indications, and I believe it's 20 patients for heme indications. And the primary reason for us recommending that is, well, there are a couple of reasons. One is so that a given protocol won't be so unwieldy that data could be accumulated at such a high rate that we aren't able to keep tabs on it from a regulatory perspective. But the other important reason primarily is because we want to be sure that the disease, the oncology experts within the FDA that are experts in the specific disease being studied have the opportunity to weigh in on development programs, because once we get beyond the 40 patient limit, then, particularly for targeted therapies that potentially have the potential to make a strong impact on cancer patients, we want to be able to provide advice about further development quickly so as not to delay the clinical development program. And so once we get to about 40 for a specific indication, we generally would like to see more specific protocols dedicated to that disease. Having said that, there are some exceptions to that 40 patient guideline, which is in the guidance. And so, if a sponsor is interested in, say, doing a small randomized phase 2 component to look at 2 different doses as part of that initial protocol, I think we'd be certainly amenable to going beyond that 40 patient limit or guideline. So I think, if you're interested in deviating beyond that, we'd be interested in hearing from you and providing additional guidance, because dose optimization is very, very important. And, we will obviously, as is mentioned, 40 patients is not very many patients to make a risk-benefit decision. And so, there are other factors that we would look into, including potentially studying additional patients for looking at markers such as response rate, for instance, or other pharmacodynamic markers that can be used to help, such as target saturation, to help make that dose decision, in terms of what to use to move forward for a more definitive trial.
That was great. I always thought of those early.
That's a guideline, not necessarily a very hard, fast rule.
Got it. I always thought of those early expansion cohorts as primarily activity cohorts, and that I understand we want to dose optimize as early as possible, but you need to know whether the drug works first before you really start to do that. This next question, you don't have to perseverate on, because I'm not sure I can even propose an answer. But if we're doing a combination study with an investigational agent and an approved agent, but for an off-label use, as in that's not what the drug was intended for, and I'm not sure why that would be happening. But how would we approach that?
My read of that question is that the question relates to if you want to use an approved drug in combination with your investigational drug in a protocol, how to justify the use of that drug, and how do you approach sort of explaining the use of that drug in your trial. That's the way I'm reading it. And generally speaking, we would expect a concise and thorough assessment of the existing published data on that drug that will be used in combination with the investigational drug, that addresses sort of the reasons for wanting to study the investigational drug in combination with this approved drug, what the potential overlapping toxicities might be, and how those toxicities will be mitigated in the trial. So I don't think I would necessarily handle it any differently than I would with two investigational drugs. Just that you have to hit all those bases and cover them in your protocol. In terms of regulating off-label use outside of a clinical trial, we don't dictate practice of medicine, so we're very aware that drugs can be used off-label in the post-market setting, for clinical purposes, patient treatment purposes.
Thanks.
Can you hear me?
I think I would have just passed. Yes, I did. It was great.
Okay.
I think I would have just passed over that one. So one of my, pet peeve, cut and paste questions is, can you just comment on the whole idea that, for 40 years we've had cytotoxic chemotherapy agents, and they had a certain mechanism of action, and we saw certain toxicities. We became used to those toxicities, and we kind of excluded them to some extent from those limiting toxicities. And now we clearly have targeted therapies with different mechanisms that have nothing to do with killing rapidly dividing cells, and yet sometimes we see, dose-limiting toxicity exceptions for drugs that are immune-mediated, and maybe just a comment on how those limiting toxicity exceptions should be put into a protocol.
Thanks, Jeff. So in general, just to give you a glimpse as to how we look at DLT criteria and protocols, we generally have kind of rules that we're looking for and what we'll accept in terms of DLT criteria exceptions and those in which we will not. And, one thing to think about is whether or not we will accept any, variability from those general grade 3, grade 4 rules is how likely adjustment or lowering of that dose will mitigate patient risk, number one. Number two, whether there are other circumstances that can mitigate the potential seriousness of any DLT that could occur, grade 3, grade 4 DLT. So, I think if there, are exceptions to the typical rule, which is that anything grade 3 or 4 will be considered a DLT, then they should be strongly justified based upon the drug's mechanism of action, how, why you think that particular toxicity should not be taken into consideration when identifying moving forward what that recommended phase 2 dose should be for future trials. We don't want to be ending up with a dose that is going to require, many dose modifications in patients, that are enrolled in subsequent trials either. So that needs to be kept in mind. And additionally, I think when thinking about the MTD, which I think is even more important in some ways than the DLT criteria, I think it's important, that investigators that are in charge of making dose escalation decisions, that they're looking into the more chronic toxicities that occur beyond cycle 1 when they're determining whether or not to dose escalate, irrespective of whether those criteria for DLT are being met. I hope I answered your question.
That actually, no, it does. It actually addresses one of the other questions with regards to how do we approach drugs with long mechanisms of action, such as immunodepression, and how we keep that DLT in. How do we consider that for DLT? And your point is that that doesn't necessarily play into the 30 days. We're not going to limit you to accruing new patients on the study, but you need to follow those and put those into your recommended phase 2 dose eventually. Here's another quick question. What are some of the most common issues that FDA identifies with INDs containing first-in-human trials? What are the clinical things where they're just kind of showstoppers from the get-go?
I think that Dr. Helms alluded to some of these already. I think that not having sufficient support for the starting dose is basically a deal breaker, and so that there needs to be quite a bit of thought and justification for the starting dose that's proposed. I think it's not too uncommon that we see very aggressive dose escalation proposals that don't take into account the steepness of the dose toxicity relationship that is observed in non-clinical trials. So, I think I mentioned this earlier, but I would be very, careful about looking at the steepness of that dose toxicity relationship, particularly if you are recommending single patient dose cohorts, for example, with, very aggressive increases in dose between cohorts. In terms of cutting and pasting issues, we, often will see some internal inconsistencies within the protocol. And so, we work hard to kind of rectify those when we think they rise to the level of a safety issue as well. Most of the issues we can work through, aside from that, from a clinical standpoint, in a protocol that's decently written. And that's through our interaction with sponsors during the 30-day process.
Thanks, Martha. Dr. Donoghue, it's been a pleasure, and I really appreciate your presentation. I think it hit all the important issues about how to develop these first-in-human trials and how to think, proactively when you're designing the trial. And that, I'm going to turn this back over to Lisa, because I hope we get to go to lunch. Lisa, let's have you jump in here.
Great. Thank you all to all of our presenters for our morning session. We look forward to the afternoon. We will